Hello friends, last studied general characters of bacteria. Today we are going to learning the cell structure of a bacteria. So usually the bacterial cell are unicellular. A typical bacterial cell shows some external and internal components. The external component comprises the capsule, slime layer, Glycocalyx, Flagella, pili, whereas the internal component of a bacteria shows the cell wall, plasma membrane, cytoplasm, ribosome, mesosomes, chromatophores, nucleoid condition of a nucleus, plasmid, etc. So look at the picture of a bacterial cell. The bacterial cell showing the slime layer capsule. So it is the outermost covering of some bacteria. It is not present in the all type of bacteria. The capsule is made up of disaccharides and polypeptides. The bacteria which possesses the capsule called as a capsulated bacteria, while those not possessing are called non-capsulated bacteria. The capsule protects the bacteria from drying and from antibacterial chemicals. It helps for immunological specificity and attached to the surface. The glycocalyx is extracellular covering which is hygroscopic and made up of the polysaccharide and polypeptide. It protects the bacteria from desiccation. The second component is the cell wall. So cell wall is the outermost layer. It is present below the capsule and it is present above the plasma membrane. It is tough and rigid. It is made up of mucopeptide, peptidoglycan, mucocomplex, muramic acid, and these all are called peptidoglycan. So peptidoglycan is made up of the unit of N-acetylmuramic acid and N-acetylglucosamine. So N-acetylmuramic acid and N-acetylglucosamine composed the peptidoglycan. Depending upon the constitutional component of a bacterial cell wall, the bacteria are divided into two types: gram positive bacteria and a gram negative bacteria. So see here the gram positive bacteria and gram negative bacteria. The gram positive bacteria, the peptidoglycan consisting 95%, then teichoic acid and tichronic acids are present, whereas in a gram negative bacteria, instead of a peptidoglycan is there, but their concentration is very less. Apart from that, it is made up of three components that are the lipopolysaccharides, phospholipids, periplasmic space and very less amount of a peptidoglycan. The gram positive is stained with crystal violet and therefore it gives the purple color, whereas the gram negative giving the pink color stain. In a gram positive bacteria, we cannot find the periplasmic space. The periplasmic space is present in the gram negative bacteria. So peptidoglycan is more in a gram positive, very less in the gram negative bacteria. Crystal violet staining is there in the gram positive. The crystal violet staining is not there in the gram negative. Periplasmic space is not in positive bacteria. Periplasmic space is present in the negative bacteria. So this is the difference between the gram positive bacteria and a gram negative bacteria. So come back to the figure. So function of a cell wall is to provide the specific size and shape to the bacteria. Cell wall protect against the osmotic pressure. Cell wall contain the bacterial antigen and lipopolysaccharide layer serves as antigenic determinant in the gram positive bacteria. The cell wall is followed by the plasma membrane. It is outermost layer of a cell cytoplasm. It is followed by the cell wall. It is present in only gram negative bacteria. The plasma membrane consisting the lipid bilayer and protein. The protein components are usually embedded within the phospholipids. So outer membrane and inner membrane are there. Outer membrane provides the barrier to the lysosome which damage the bacteria. The outer membrane is important role in the cell division and sporulation. It is act as an endotoxin. Outer membrane is permeable to the small molecules. Inner membrane is selectively permeable, means some molecules comes inside, some molecules go outside. It contains the inner membrane contains the permease enzyme and it is a site of a electron transfer system. The next the component is the cell cytoplasm. So bacterial cell cytoplasm is the colloidal in nature. It's showing the movement that is called as cyclosis. The cell organelles like ribosome, mesosomes are present in the cytoplasm. The other cell organelles like mitochondria, chloroplast, endoplasmic reticulum, lysosomes are totally absent in the bacterial cell. So ribosome present in the bacterial cell is 70s ribosome. It is made up of 50s larger subunit and 30s smaller subunit. The next is the mesosome. So look at the mesosome. So infolding a plasma membrane in the cell cytoplasm called as a mesosome. It is also called as chondroids. They are made up of tubules and lamellar whorls. The mesosomes are of two types, septal mesosome and lateral mesosome. Function, it involved in a DNA segregation. It involved in the formation of septum. It involved in a export of extracellular enzymes. It provides the binding site for the chromosomes and plasma, the plasmids at the time of replication. It involved in the DNA replication. Then chromatophores are there. That chromatophores you are not looking there in the bacterial cell. These are the color pigments, and due to that the bacteria getting colors. So chromatophores are the pigment present in the membrane and they are of various colors, but green colored pigments are helping in the process of photosynthesis and they are present in the cyanobacteria. Next, magnetosomes. Magnetosomes are also not there in the figure. These are the cellular inclusions present in the some bacteria. They are present in uh, some bacteria, not in the all. They are responsible for orientation of the bacteria in the magnetic field. Means what? The bacteria fight against the magnetic field. Sometimes bacteria go away from the magnetic field. Some bacteria attract toward the magnetic field. So such a attraction to the magnetic field is due to the magnetosomes. Then very important part is the nucleus is primitive. Nucleus does not having nuclear membrane, nuclear pore complex, nucleoplasm, nucleoli. Nucleus is a very primitive one, earlier not developed, not advanced nucleus. That nucleus is called as a nucleoid. So in the bacteria, the nucleoid is present, and bacterial cell possesses some extra chromosome that are called as a plasmids. The plasmids are many plasmids, 2 to 100 plasmids are there in the bacteria. They are called as a episomes. Then look at the right hand. The bacterial cell possesses the flagella. So the flagella are useful for movement of the bacteria. It possesses in the motile bacteria. In some motile bacteria, the flagella are present. These are the long and branched thin helical appendages called as a flagella. Now the flagella are of various types: polar flagella, non-polar flagella. The polar flagella means the flagella is present at the one end. They are called as a polar flagella. So here the flagella is polar flagella. uh, Only one flagella present, that called as a monotrichous. If both pole having a flagella called as a amphitrichous. If two flagella are present at the one pole called as a cephalotrichous, and many flagella are present at the both end called as a lopitrichous. If flagella is not present, that are called as a atrichous bacteria. The non-polar flagella or non-polar bacteria are the total body is covered with a flagella that is called as a peritrichous flagella. So that is present in the clostridium. Now let's see the example. Monotrichous flagella we can report in a vibrio. Amphitrichous report in a nitrosomonas. Cephalotrichous reported in a Pseudomonas, whereas the Lopitrichus report there in the Spiralium. So, under the electron microscope, such a flagellar arrangement can be studied. And last point is the fimbriae, also called as pili. These are the short hair-like appendages arises from the surface of a bacteria. So the term fimbriae was firstly coined by the scientist Dugid and associate, and the term pili coined by the scientist Brinton in 1959. So the fimbriae are observed mostly in the gram-negative bacteria and are made up of a protein fimbriae or pilin protein. So they are also helping in a movement or the movement or the motile nature of the bacteria. So this is the structure of a bacteria that we have studied here. So cell structure of a bacteria we have studied. You can be look very important point for the examination. You will get the some MCQs, that is gram positive bacteria's composition, then your flagella's position. On that basis, the monotrichous type of flagella is studied in dash dash bacteria. The answer is vibrio. Then amphitrichus is studied in dash dash bacteria. That is answer is the nitrosomonas, like that way. Peritrichus is found there in the clostridium. So this is all regarding this topic, and we are stopping now. The point cell structure of a bacteria. Thank you very much.